This week, we are spending the entire week on talking about DNA, the DNA molecule. It's only 2 pages in the textbook, page 101 and 102. Here we go. And this is what we did yesterday, reading page 101 and 102, and questions 2A, B, C. This is all about how DNA makes an exact copy. And then today we're going to make an actual physical model of DNA with colored paper and pipe cleaners to make a 3-dimensional model. Then tomorrow we're going to do an extraction so you can see real DNA. These two things together will give us lots of information about DNA. So let's look at the questions from yesterday. Whoops, that's the quiz answers from last week. So 2A said list the nitrogen bases. And so the nitrogen bases, you can find that right there in the reading, right? What are the nitrogen bases of DNA? Yes. Adenine, guanine, thymine, and cytosine. Okay? Then number 2 says which one matches with which, basically. They say it in a much more confusing way, but A always matches, A always matches with T, adenine always matches with thymine, and cytosine always matches with guanine. Now, the way I always remember, I always have little ways to remember, right? The way I always remember, look, A and T, they're both kind of pointy letters with straight lines, right? All straight lines. The 2 straight line letters match up. And then C and G are both curvy letters. The curvy letters match up. Okay? Oh, yeah. And in real life and in your model, there's a puzzle piece kind of fit. It's actually, partly chemical in the way the molecule is shaped. But for us, when we make our molecule today, you're going to actually make it so only A can fit with T and only C can fit with G. Because that shows, this is going to be why it's so important, only one matches with its particular mate. And because of that, when the DNA unzips and copies itself, it has to be an exact copy. Okay? So I know during second period when I was helping some of the kids with this answer, they were like, I don't understand that last one. It's so confusing. This right here, it says, if this is the original, AGATTC, right? It says, what does the other side look like? And people are like, well, how in the world am I supposed to know that? It's actually pretty simple, right? Right? Because A only matches with T, and G only matches with C, and then A matches with, and T matches with, and A matches with, and C matches with. So that's what the other side of the DNA strand looks like. Okay? And it turns out, I will ask you this question over and over, just kind of drill it into your head. It's very easy. It looks complicated. It's very easy. And then the other question I put there, which is also on your worksheet for today, is how in the world does DNA make an exact copy of tens of thousands of base pairs? And the way I like to describe it is a 3-part process where it untwists, unzips, and then builds a new side. So here's the cool thing. If you do this in your notes, it's actually really easy to draw a molecule of DNA. Just make a squiggly line, and then make another squiggly line, and then it's unzipping here. So how does DNA make an exact copy? It untwists. You could put a little star next to this question, because you know what? This question's on the unit test. The unit test in December. Untwist, unzip, and then rebuild. And so if we just kind of drew in, we're just going to make a random thing there. And in real life, the order of the base pairs codes for certain things. But I don't happen to know any of those off the top of my head. So for this, what would the other side be on the original molecule? This would be a T. This would be a. This would be. This would be. Okay. So it unzips a special little. It goes around. It unzips it, and so now you only have half. Thank you. And what happens then is some little ribosomes come and build up the new side. So take a look here. What's it going to build onto here? What letter is going to build onto here? Okay, somebody else. What is going to build onto this one? A. What's going to go on here? Everybody, what goes on this one? And this one? This one? Okay. And then on this side, what goes here? Here? Okay. And then it'll retwist back up and you have two identical molecules of DNA. Piece of cake? Oh, excellent. So today we're going to build a molecule. I have a worksheet for you and some supplies for you. And then you're going to answer some questions, and guess what? The questions are going to be these same kind of questions. Okay? Excellent. You now have your Make a DNA Molecule worksheet. Put your name on it. And what's going to happen here is by the end of tomorrow, you will need to have answered all of those questions on the back. The questions on the back, 1 through 9. Questions on the back, every single answer is on the front side. So as you read over this, as you read over this, all the answers are here. Everybody is going to make their own DNA molecule. Like any lab or activity, you can help each other out at your table group, but everybody will do this part today and tomorrow. If you don't finish today and tomorrow, then you'll need to do it in class. Now, what you are going to make is a DNA molecule. So if you look at this on the front, it talks about using string. Special for you guys, instead of string, this year I bought some pipe cleaners. Due to the craziness with pipe cleaners last week, I will portion out the pipe cleaners. Everybody gets 2 pipe cleaners, no more, no less. Okay? Then when you're making this, there are detailed directions here. You're going to make a model, and as you see here, you need to make sure on your model that this only fits with this, and the other one only fits with the other one. So you'll see this. I'll show you the example for how to do it. This will be due at the end of class on Wednesday. If you do not finish it, then it will be homework.